thank you very much, Russell, for agreeing to give a Build a Cell seminar. And you can get started.
All right, thank you. I really appreciate the invitation. I'm enjoying the opportunity to talk to this group, and I hope to hear what you think about what I have to say. So I am a computational biologist. I'm head of the Ray and Stephanie Lane Computational Biology Department at Carnegie Mellon, as well as a professor of biological sciences. I did want to say that I've had a circuitous route through research in computational biology. I started as an undergrad with a little sequence analysis, but mostly working on problems in biophysical modeling and simulation. Continued that through grad school, went to do a postdoc in a wet lab biology lab where I got more into problems of kind of big data mining, the early days of when there were big enough databases to start doing data mining as a form of research. I wanted something different after that, and I went to a company, Cellular Genomics, that was competing to sequence the human genome for the first time. Got into other problems and more classic algorithmics and algorithms for sequencing, of course, and sequence assembly, but also population genetics and phylogenetics as I got into kind of questions of genetic variation and population variation. And then I moved to Carnegie Mellon, where I've tried to combine these different agendas in computational genetics, genomics, biophysics, and so forth. And for the past really about 10 years now, my lab has shifted into primarily doing cancer research. And that's where I'm coming from, the perspective of someone who has done a little work in synthetic biology and other related areas. I used to do a lot more in biophysical modeling, for example, but is now mostly working on cancer research. I wanted to share a little bit about what I think cancer has to teach anyone who's interested in creating a cell or working in the synthetic biology of creating an artificial cell. And I'll just say right now what my key points are, and then I'll kind of walk you through where I'm coming from there. One of these is that I think you have to keep in mind that cells are inherently dynamic systems, and they're constantly in flux from both intrinsic forces, so genetic evolution of your cells, as well as intrinsic forces, so influences from outside them. The second thing is that cells don't always do what you want. They're going to evolve whether you want them to evolve or not, and their behavior is going to be highly stochastic, but that's not to say it's entirely unpredictable. And I think this leads to some conclusions. I would suggest that robustness to this is something you have to keep in mind if you're going to create a synthetic cell that's going to continue to be useful for a persistent amount of time, that you have to account for the stochastic behavior and robustness to it. And the third thing I would say that we learn quite a bit in cancer that I think is a useful lesson for anyone trying to build an artificial cell is to remember that cells don't function in isolation. They exist in an environment and generally as part of a community of other cells. And this has become quite important in how we understand cancer. And I would argue is also going to be something that I think anyone working in synthetic biology will want to keep in mind. So just to move from there, I got into cancer research from the perspective of someone starting in other kinds of evolutionary research. So working on cancer from the perspective of a disease of evolution. So cancer is governed by evolutionary forces of diversification and selection. We believe that cancer usually is beginning early on with a genetic mutation that is often a mutation that promotes further mutability, so damage to cell repair, replication machinery that leads to a proliferation of heterogeneity in the cell population. Cells naturally develop genetic heterogeneity, but they have mechanisms to limit this and prevent this to some degree. And when those break down, you get a lot of cell-to-cell heterogeneity, which creates opportunities for selection. And what we think of as cancer is really the phenotype of selection for the more aggressive clones, or ones that are better escaping different controls on cell growth. So this has led to, this observation has led to an entire field of tumor phylogenetics that my lab and now many others are working in. And some of what I want to tell you about in this talk are some of the lessons we learned from working in this field of building evolutionary models at the single cell level from cancers. My own lab is largely methods development focused, so we're working on how you ask questions about the evolution of a cancer. How do we reconstruct evolution of cell lineages? And we've been working on that since pre-genomic methods, so essentially methods that could be conducted back before it was possible to sequence whole tumors, or at least practical to sequence them at any kind of scale, to when bulk genomics became available, essentially being able to sequence a sample of many cells and get a kind of average profile, and moving into the single cell era. And today, increasingly working with multi-omic combinations of data. So this is just a slide illustrating a method that is combining all of these different data modalities, so bulk sequencing, single cell, and a pre-genomic technology, MyFish, which is basically counting fluorescent probes for particular regions of cells. And what this is essentially meant to illustrate is that much of the action in this field is taking these continuing advances in technology and trying to figure out how to leverage them better to learn more and more about what's happening at the single cell level. And for the purposes of this talk, I don't want to dwell on how exactly we do that, just to say that the biotechnology is constantly improving and the computational biology is figuring out how to use that more and more effectively to learn in very fine detail what happens as cells evolve and how the many mechanisms by which this occurs. There are whole other dimensions of improvement based on our understanding of the biology. So in addition to the biotechnology for profiling what's happening at a cellular level getting better, we understand more and more every year about the biology of the ways that cell lineages evolve in cancers as well as in other contexts. So we know that, for example, there are many different kinds of genetic mutations that can occur at a single cell level, and pretty much anything you can think of that isn't going to kill the cell is going to happen sometimes in tumor cells and be part of the portrait of how tumor cells evolve. So this is an illustration of a method for taking point mutations, which is what a lot of classic evolutionary theory involves, what are called single nucleotide variants, or SNVs, and combining them with other kinds of variations, structural variations, which describe large-scale gains, losses, and rearrangements of the genome, copy number alterations, which describe gain or loss of large pieces of DNA in a genome, and putting all of these together. And the more we understand about the biology behind how this happens, the more complete a picture we can make about what's happening in these cell lineages, understanding how these different kinds of variants interact with one another and how they influence one another, or in some cases confound one another in our interpretation. These are things we're also bringing to the single cell level and really starting to see more and more examples of how the different kinds of mechanisms of mutability interplay in cancer evolution. So different cell lineages hitting the same regulatory pathways from different combinations of mutation types, for example. And so over time, we're developing a more and more accurate and detailed picture of how tumors evolve. And I'll say that one of the big conclusions from this, the more you look at these kinds of phylogenies, the more you realize how idiosyncratic and stochastic it is. Any two cancers that you look at are going to be doing different things in their evolution. Any two cell lineages you look at in the same cancer are going to be doing somewhat different things in their evolution. And this is an important point to keep in mind. Evolution is a highly stochastic process. There isn't one trajectory or one solution to an evolutionary problem, and different cells will go different ways. And I'll say more about that a bit later. The technology is continuing to evolve, and there are lots of exciting directions. This one may be less relevant to synthetic biology, but is quite a big thing in cancer biology, and that is blood-based tumor profiling, what's called liquid biopsy, which essentially means profiling what's happening in tumor evolution based on looking at DNA or in some cases, cells shed into the bloodstream from a tumor. This allows you to start creating not just a portrait of evolution as a static evolutionary tree, but understanding the population dynamics of the different clones in a cancer. So this isn't just a linear sequential process where cells are in one state and then later they're in another state and later they're in another one. cell populations establish heterogeneity with different combinations of cell types, with different combinations of genetic mutations, and these undergo their own kind of population level evolution. So what this diagram at the right here is showing is a reconstruction from some data from a large consortium called TracerX that's doing a lot of this blood-based sequencing. And essentially what we've done is use their data to build an evolutionary tree of clones in a tumor and then track how those clones changed over the course of treatment for one patient. So this is showing how at the beginning, so essentially after the tumor is removed, you don't see a lot of activity of any of the clones. You can see some residual level. And you can see over time that one of those clones is really starting to grow. That takes off quite dramatically. So this little amount of growth you see after 91 days turns into a large recurrence by 174 days. At this point, 2 of the clones have grown quite dramatically, another one to a lesser degree, and that would lead to a change in treatment as the doctors are trying to get this relapsing tumor under control. Now, usually they would be applying a therapy that's targeted to the majority of what you see in the tumor, and that's probably what happened here. They gave therapies that were very well designed for those dominant clones in red and orange, and those clones respond and fall off to those, but other rarer clones are resistant to that therapy and grow to replace the dominant ones and give you a resistant tumor that's no longer going to respond to that therapy. That unfortunately is a very common outcome of cancer treatment. But what all of this is saying for the purposes of this talk is that we're gathering other dimensions of data, understanding how this evolution happens, not just at the scale of mutations in single cells, but dynamics of populations established by that process of mutability. There are other directions in this. They're quite exciting in cancer research right now. So many people in the field are very interested in bringing in the spatial dimension, using new technologies for spatial genomics, spatial transcriptomics, for example. But my lab is kind of doing our own thing there with a collaborator, Lian Zhao, using very high resolution fluorescent imaging combined with DNA sequencing as a way of trying to identify what are the major clones in a tumor, where are they occurring, in a way that we can map them to very high resolution spatial images. But basically what this is saying is that we're gaining another dimension of what's happening in these processes as we can start to see structure of a tumor in terms of these clones not just being well mixed in a solution, but establishing in different niches, different spatial environments, different combinations of clones and populations of these, as well as what's not visualized here, populations of other kinds of tumor cells. And what all this is to say is that there's a constantly changing landscape of advancing biotechnology, biological knowledge, and computational biology that is showing us in ever more detail what happens in a population of somatically evolving cells. And really the conclusion I would like to draw for the first part of this talk that I would take to anyone interested in cell biology is to remember that a cell is not a static entity. Cells evolve, cells, even our own cells that have quite a few mechanisms built into them or evolved into them to prevent them from evolving, continue to evolve somatically, and they establish a heterogeneous evolving system of populations of cells that itself is heterogeneous in both time and space. And I think this is an important thing to keep in mind if you're going to be creating a synthetic cell. If you create something that is capable of self-replication, that is really alive and reproducing in that sense, eventually evolution wins out and it's going to do something like this. So it's important to keep in mind what that means and what mechanisms you have for shaping that. And that kind of leads into the second thing which I wanted to talk about, which is getting into a different kind of methodology we apply in this field, and that is machine learning. So these methods for tumor phylogenetics I was telling you about are largely about looking to the past. So being able to look at a tumor and say, what do we think happened to get it to that point in time, and reconstructing that process of evolution. But a lot of what we're doing is also about looking to the future, saying, based on what we can observe of the process of evolution, what's likely to happen next. Now, as I mentioned, these are highly stochastic systems. If you look at two different tumors that look the same, you're likely to find that they do very different things. If you look at two different cell lineages in the same tumor, they may do very different things. And another way of saying that is that there is an inherent randomness, and this is not going to be entirely a predictable process. You cannot just look at a population of cells and say with any certainty what's going to happen next. But we want to be able to ask more rigorously, to what degree can we predict this and what drives those predictions? So this is illustrating a kind of very simple machine learning study where we were using a sort of ablation analysis. So taking different kinds of data and trying to hide subsets of the data to get at the question of how much is predictable about the future evolution of a tumor and how much of that is predictable just from understanding the process of evolution acting on it. So what we did was take these evolutionary reconstructions under a variety of different conditions, different tumor types, different data types, different ways of building the tree, as well as different outcomes that we're looking at, take different ways of quantifying the evolutionary process, combine this with other information we have about the tumors, and ask under different combinations of these data subsets, how predictable is the process? Or another way of saying that is, how much of our predictive power for the future of a tumor do we get just from the evolutionary process relative to our total predictive power? We can find that there are lots of things about a cancer that are predictive of what it will do in the future, some of these being things that describe the evolutionary process. Some are more prone to point mutations, some to copy number changes, some are more prone to particular kinds of point mutations. GCC to GGC versus GTG to GGG is predictive, whether they have large or small copy number changes, whether they gain or lose, as well as higher level features like do they tend to build very branchy evolutionary trees or very linear evolutionary trees? All these things have some predictive power for what's going to happen in the future. So essentially, there are different ways of evolving, and it matters which ones you pick. It's a complicated question how it matters. So this is trying to illustrate some of the complexity of the high-dimensional space of evolutionary phenotype. So if we take all of these different features, you can kind of plot them. This is T-SNE plots, so basically two-dimensional projections of this high-dimensional space of evolutionary phenotypes. And you can sort of see there are these complicated manifolds. There are regions that correspond to good and bad outcomes. But the point is that there's a lot going on here, and we really don't have a full handle on it yet. But at a very high level, we wanted to try to answer this question: how much can you predict from the way a tumor is evolving, what it's going to do in the future? And this is kind of a summary plot of that, trying to quantify this in terms of the amount of hazard we're able to account for with different sets of features. And essentially the high level conclusion here across many different ways of asking this question is that we can predict about a third of the hazard of future progression of cancers from the evolutionary feature, so how it's generating evolutionary novelty. And I'd argue that it's not so important that it's a third versus a fourth or a half, but it's important that it's not zero and it's not 100%. Essentially, this is saying there are things you can predict, but there's a lot you can't. But there are things that are predictable there. And I'd argue in the context of synthetic biology, predictable means potentially actionable. You're not going to be able to get away with self-replicating cells not evolving at all, but they can evolve in different ways. And I think this is a space of creating synthetic biology that is worth considering. How do you pick the ways you control evolution or the kinds of evolution you'll allow versus not allowing to at least help shape things in the direction you want them to go? We're continuing to work on this question, trying to understand how those profiles evolve over time. So how your ability to predict the future of a tumor evolves over the course of history of that particular tumor, as well as bring in other kinds of data. For example, when I mentioned that we're mapping to spatial data, part of the reason for that is so we can bring in much finer scale information that you can get from high resolution imaging and start to ask, Are there features of the images that have predictive power? And the short answer is that there is. There are things about a cell that tell you about its future that you can see visually, things about relationships between cells and communities of cells that have predictive power. And then this is early stage work. I don't want to draw any sharp conclusions from it other than that all of these things have predictive information about what's going to happen in the future and contribute to this picture that there are things we can understand and potentially control about the evolutionary process. You can't necessarily suppress it, but there are ways of shaping it that I think are worth keeping in mind. And that kind of brings me to the second conclusion of this talk, which is that single cell evolution is a highly stochastic process, but it follows predictable trends, and we are coming to understand these. And through interpretable machine learning methods, we're starting to gain an idea for what's important in shaping those evolutionary trajectories that I think potentially could be controlled. There's a quote from Mark Twain that I think is very relevant to cancer here. So he said, "History doesn't repeat itself, but it often rhymes." And I think that's a good conclusion about single cell evolution as well. No two single cells are going to evolve in exactly the same way, but there are trends and you can get a handle on them. But there's a whole other piece of this process that I haven't gotten into yet that you can't see just from looking at what's going on in individual single cells. And this requires bringing in other sorts of methodologies. So in my lab, we've been looking a lot at RNA sequencing as well as the DNA sequencing. Many others are looking at this as well as things such as epigenetics, chromatin structure. Pretty much anything you can look at a cell can be a readout of a process of evolution and will tell you something about how cancers are evolving. In the RNA-seq, what we wanted to understand is what evolution of cell populations looks like in terms of patterns of expression we see in the cells. We were particularly interested in this in terms of the cancer evolution. So understanding, for example, evolution from a primary to a metastatic site within a tumor. And you can use similar kinds of methods to what we use for the DNA to reconstruct this process. You need to know a lot more about the biology to get a coherent picture out of this. If you just look at isolated genes and try to build these evolutionary trees, it's often kind of a mess. You can't really figure out what's going on. But by knowing that genes act in terms of modules and pathways, common regulation for performing common function, by recognizing these gene expression signals as signals of activity or disactivity of different pathways, we can get a much more coherent picture of the evolution and start to reconstruct what's happening in terms of expression evolution, which is in some sense a proxy for functional evolution of cell populations, in this case in cancers, and start to see again some recurring patterns as we go from primary to metastatic tumors, particular kinds of pathways that tend to be differentially activated or deactivated in particular ways for particular kinds of transitions. And we can bring that as well into pseudo-phylogenetic models of tumor evolution. So just as you can build these models where we can look at these states of DNA evolution, how they evolve in a kind of tree and propagate and mix over time in a tumor, you can talk about states of expression evolution. And I'm calling these pseudo-phylogenies because they're not proper phylogenies, because what we're seeing here is not really the evolution of single cells. It's actually conflating a number of different influences. So we can look at these trees describing these kinds of patterns of activity that propagate across a tumor, and we can see how those correspond to increases and decreases in activity of different pathways. But it's important to remember that what we're seeing here is really a mixture of, in part, changes of expression patterns in the tumor cells themselves. So tumor cells, as they evolve, they get mutations which may change their activities and lead to direct functional effects, what we would call cell intrinsic evolution. But we're also seeing the influence of other cells around the tumors. So tumor cells occupy communities in which those communities may contain one or more kinds of tumor cells that may have different functions, as well as non-tumor cells of various kinds. Some kinds of clones within a tumor may like to be associated with a particular kind of healthy cell. Some of them may establish communities with other particular kinds of tumor cells. Some of them really need to be near a blood vessel, let's say, whereas others are fine far from a blood vessel. And you get interaction with recruitment of other cell types as well. Some tumor cells are very immunogenic. They recruit immune cells to their communities. Others evolve ways to be invisible to immune cells and exclude them from their communities. And what we're seeing here is in part a combination of that as well. So when we talk about a state of expression, we're seeing a state that's established by both changes in the activity of the tumor cells and changes in the composition of the community around them in terms of the other cell types associated with them. There's yet another layer of effect there in terms of patterns of expression in those individual cells, because cells influence one another's behavior. A tumor cell will change the behavior of the healthy cells around it, change their expression. The healthy cells around a tumor will change its patterns of expression in quite a complicated feedback. If there are immune cells present, they will behave differently in the vicinity of the tumor than they'd behave if they were in another environment. And all this is influenced not just by the cells, but by the environment in which they find themselves. Once again, technology can give us better ways of understanding what's happening here. So bringing in some of the newer single cell technologies, we can use similar sorts of methods to rationalize what's going on more accurately at the single cell level and start to take apart some of these confounding effects to understand, for example, what exactly is changing in the populations of cells around the tumor cells at different sites, and separate that from the influences of expression changes in single cells. So to understand that in some sites, maybe you have few fibroblasts and in others lots of them, as well as that those fibroblasts may be changing their expression in one site versus another site, and influences in the expression of the tumor cells in one site versus another site. So we're getting a better and better picture of what's going on, but this is a very active area of research for us and for others in the field to try to tease apart all of these different influences. We're trying to get more of an idea what's going on there by bringing in ways of integrating these different portraits that we're seeing from these different sorts of technologies. Just as bringing in different modalities of DNA sequencing is increasingly important in taking apart the different pieces of what's happening here. Likewise, bringing together these portraits we see from looking at the tumor DNA versus looking at the RNA that's reflecting on both tumor and non-tumor cells. This is increasingly important to understanding what's going on. One way we're trying to do that is by using the fact that RNA data can give you some limited information about what's happening in the DNA. You can see some mutations in the RNA. You can, to some degree, identify large-scale copy number changes in the RNA. It's much sparser and noisier data than you get from looking at DNA directly, but that's one way of trying to get at, on a common platform, what's happening in terms of both expression and mutability. But really what we'd like to do is get a more complete picture of everything we can learn from both of these kinds of data sets. And this, again, is revealing ever more complexity to what's going on in these systems. So this is an example of what we get when we use some data, not our own data from the literature, Ho et al., of where there is DNA and RNA information at the single cell level available from the same sets of single cells. And we can start to put together a phylogeny from the DNA and map the RNA to it. And one of the conclusions we can draw is that a lot of it does not by itself make sense. That once again, you can't just look at the tumor cells. You can find that there are some genes that have coordinate patterns of expression that correspond to the DNA-based phylogeny. So this ARID1A gene, for example, that has high expression in one clade and low expression in another clade, that very likely corresponds to a direct effect of mutability on expression of that gene. But for the most part, looking at important driver genes, in this case in liver cancer, you don't see any coordinate pattern. What's going on may be just random chance. It may be indirect effects through, for example, a mutation elsewhere in the genome that's recruiting other cells that are communicating with these cells that are influencing expression and so on. Where you do see coordinate change, it often is because of copy number changes. So this is showing how we do see a region of multiple genes that are showing coordinate change in expression, well consistent with the clade structure of the phylogeny, but it's because of large-scale copy number changes that are defining both the clade and the expression pattern, essentially a large segmental loss of a piece of chromosome that creates a split in the evolution of the tumor and also creates identifiable expression patterns. So the overall picture is going to turn out to be some combination of these sorts of direct effects. A copy number change leads to an expression change, which leads to functional changes in the tumor cells, and more indirect effects. And what all this is doing is leading us to what I would describe as at least approaching a more unified view of clonal evolution. And this is a view of clonal evolution in cancers, but I would argue the same picture is going to apply to any kind of cell you talk about, synthetic or real. Your cells are going to have genetic variation. There's going to be a process of clonal evolution, establishing distinct cell populations, establishing heterogeneity among those. And this is one piece of the evolutionary process, but that's layered on top of a population level dynamics that's going on there, where these different clones aren't simply replacing one another, but often interacting, coexisting in the same environment and creating possibilities for selection or adaptability as that environment changes. That's layered on top of spatial dynamics of different environments, in this case, different environments a tumor might occupy, such as a primary site or metastatic environments, or even different local regions within one of those sites. But in synthetic biology, this could be any number of different niches that your cells might encounter when you put them to work for whatever you're designing them for. And that in turn is interacting with regulatory changes in both the tumor cells and the non-tumor cells with which they're associated through a complicated process of cell-cell communication that others in the field are working to understand now, and that we're only really beginning to get a handle on. So I would argue that in the cancer case, this is a hard problem in part because you can't really understand any piece of this without understanding how all the other pieces are influencing it. And I would say the same thing is going to be true in synthetic cells. All of these different effects are going to be acting on your synthetic cell systems, and you're going to want to have ways for accounting for that if you want to be able to build cells that are going to be robust and at least for a good amount of time are going to keep doing what you want them to be doing. So that kind of leads to my third conclusion here, that a cell doesn't exist in isolation. It's normally part of a community of cells in a microenvironment that both shape and are shaped by it. So there are lots of other things I could be telling you about that my lab is doing and others in the field are doing. I know the synthetic biology community has been really ahead of the curve in a lot of ways on first principles modeling. So trying to say, from what we've designed, what do we think it's going to do? And this kind of dynamic modeling I haven't talked about here, but I think it's another part of the puzzle of how we make sense of all of these kinds of information. So within the cancer community, there are kind of mathematical oncologists who do a lot of this sort of dynamic first principles modeling, and computational cancer biologists who do more kind of data analysis, often from simpler models, because that's traditionally what we could fit to data. But I think these are really converging, as you really need more complex models of the biology to make sense of the data. And at the same time, you want to use the data you have to constrain these models better. So I think there's a lot more to be done bringing those fields together. I think there's a whole space of study design that is underappreciated right now in genomics work, and I would argue probably in synthetic biology. I'm sure some of you know better than I do, but as we get more and more into understanding how all of these different factors interact in producing a particular kind of behavior, it becomes increasingly important to bring in different modalities of data. And as I hope I've convinced you, each of these is giving you different windows, and it's really only when you start to combine all of those windows on the process that you really begin to get a handle on what's going on. But that also creates a very hard problem of what data do you look at and it matters quite a bit how you make that selection. I'd argue that there are hard problems for optimization and modeling to be solved there about the future of just how do you profile a population of cells if you want to understand its evolutionary process. And the final thing that we're working on, many people in the field are working on, is the whole AI and machine learning direction here. I didn't say too much about that other than that we're kind of using it here, but there are a lot of questions in how do we better predict the future of a cell system? How do we predict how it responds to interventions? For example, if you apply a drug, what's likely to happen to one tumor versus another, as well as using interpretable AI to start to take apart the picture of what is actually driving these changes. This is an area that I think AI is taking over a great many fields of computational biology and biological modeling. Again, some of you probably know more than I do, but I'd be willing to bet there's a big future for AI in creating predictive models of synthetic cells and using that in the cell design process. So AI-driven models are really supplementing or replacing first principles models in a lot of domains because of their power for really taking account of a lot of unpredictable factors, very high dimensional data sets. And I'd be willing to bet there is a lot of interesting work comparable to the cancer work to be done in synthetic biology in coming up with the predictive models that you can use as a tool in assisting design and developing cells that are going to do what you want them to be doing. So just to summarize here, I'd like to say that three key points that I draw from the cancer research that I think will apply to any cell population and hopefully give you some useful guidance for designing a cell is, first of all, that a cell is not a static entity. It's going to be a heterogeneous evolving system that is heterogeneous in time and space. That evolution is a highly stochastic but somewhat predictable process. And understanding this process requires understanding that cells don't exist in isolation. They're parts of communities and environments, and they influence those communities and environments and are influenced by them. And these are things I would argue you need to be accounting for if you're going to be manipulating an existing single cell or trying to create a minimal cell. So if you want to create a minimal cell, you need to consider that even a minimal cell has to be robust to both intrinsic and extrinsic forces that are acting on it. And these are things that I would say are lessons from cancer that I hope provide some useful guidance for synthetic biology as well. I'll just close with some acknowledgements that what I described here was work of many people over the years in my own group, labs of collaborators at Carnegie Mellon, experimental and clinical collaborators elsewhere, as well as funders, sources and sources of data. And I thank all of them, and I thank all of you for your attention. I'd be happy to take questions with any time we have left.
Thank you. Thank you so much. We have a lot of questions in chat. That was a great talk, and I have a few questions myself, although I see some in chat similar
Okay,
to what
thanks.
I'm interested in.
Yeah.
Can you read a question in chat out loud that's going to go for the recording?
Yes, I couldn't follow it in real time, but I'll read what I'm seeing now. All right, so from Charles Rumberger, we had, and to make it worse, many cell lines are accidentally HeLa now. And then Kate, you said, wait, how HeLa contaminated other cell lines? Yes, we saw something like that in our lab. It's weird. So I'm not sure what to say to that, except it sounds very interesting to me.
Yep, I think next one's a comment too, and then the questions start.
Okay. Yes, maybe I'll skip ahead to the questions. Okay, so from Angelica, Russell, great talk. When you do single-cell-seq for lineage tracing, do you separate information about organelles, for example, mitochondria variances across cell lines? I would say in my lab, we haven't. I don't know if anyone else in the field has been looking at cancer cell lineage tracing from the point of view of mitochondrial evolution. It's an interesting question, and it's a harder kind of evolution to track because you may have uneven sharing of mitochondria, and you have heterogeneity of mitochondria even at the single cell level, and in cell division they may be unevenly partitioned. So we haven't looked at that. I'd be surprised if that wasn't part of the picture of what's going on here. I don't know if someone else has looked at it. It's a very interesting question. Let's see, from Emmanuel Ruse, I have a question related to your first conclusion. Do you think self-replication is necessary for this kind of unpredictable evolution? What if we have cells that grow but need some trigger to replicate? They still, in theory, could evolve as long as there's mutagenesis, right? Yeah, so it's an interesting question, but I would argue that I think you're right there. Even if the cells need a trigger to replicate, if they're replicating, there's going to be some rate of lossiness in that process, some rate of error. And if you have errors over successive generations, you're creating opportunity for heterogeneity, which creates opportunity for mutagenesis. One of the things you observe with cancers is that they are very clever in a sense, and I know it's not good to anthropomorphize, but they act as if they are very clever at escaping anything you try to do to get a handle on the evolutionary process. And in the cancer case, that it has very important implications because it's all about drug resistance and cells, cancers escaping therapeutics. But I think it's a general property of evolutionary systems. If you try to stop evolution, but the potential is there, sooner or later, eventually, given enough time and enough generations, something is going to break. However, you are stopping evolution, and they'll evolve a way to give themselves the trigger to replicate, or something like that, and the process becomes out of your control. So I do think that is right. As long as you've got mutagenesis, at least in theory, over long enough time scales, there's an opportunity for evolution. Let's see, from Paul Reich, would you be able to estimate how complex a genome is needed for the stochasticity of evolution to kick in? In synthetic cells, we now have tiny genomes, no more than a few dozen genes. How big does it need to get to start seeing some emergent evolutionary effect? My own take would be that you can't avoid the stochasticity by having a small genome. And some viruses have very small genomes, and they are quite rapidly evolving and quite capable of similar sorts of evolutionary innovation, like escaping therapeutics or evolving to escape the immune system. So I do think that a small genome is not necessarily going to protect you. In some ways, that's part of my point in bringing this up in the context of minimal cells, is that you actually need some complexity to keep the evolutionary process under control. So our own cells have a lot of mechanisms designed to, for example, reduce error rates in cell replication, add additional layers of proofreading, add ways to kill a cell if it's showing signs that it might have mutated in a bad way. So there's a lot of machinery to keep that under control. And I think being a smaller cell or a smaller genome may actually create more risk by not having some of those checks that we've evolved to prevent evolution from occurring. Let's see, next from Peter Hahn. When communities of cancer cells coordinate, is it small molecule mediated, or they actually exchange genomes or interact in some other ways? I mean, it could be a lot of these. So there are well-defined things such as cancers will have cell surface receptors like growth hormone receptors, and they may evolve to overproduce those growth hormones as well as to be oversensitive to those growth hormones. So they are coordinating through small molecules such as that. They're coordinating through immune interaction. So the whole portrait of what the immune system is doing in cancers is a very active area of research where they're largely communicating through these similar kinds of small molecule signals, and the tumors may be interfering with those signals in different ways or evolving ways not to respond the way a healthy cell should to some of those signals. So I think that is part of the process. Exchanging genomes, and this is an interesting question because I'm not sure if it's ever been observed that cancers exchange genomes, and you can get some things like cell fusion where there would be the possibility that you're pooling different genomes and getting some hybrid among them. I don't know if there's anything like bacterial horizontal transfer. I guess I would say, from having worked in cancer biology for a number of years, I wouldn't be surprised, but I don't know if there's actual evidence for that. But there are a lot of other ways they can interact. I mean, some of the interesting things you get in cancer is that some of the most aggressive cancers have a high propensity for duplicating their genomes, and many cancers have propensities for aneuploidy, so creating extra copies of chromosomes. So you can get an evolution within single cells of a kind you might have a hard time seeing in a single haploid organism, because they really have multiple copies of the genome. And I kind of thought in a theoretical sense that some of the things, there's a lot of evolutionary theory about why, for example, you need sexual reproduction. You end up with evolutionary dead ends if you just have mitosis and no meiosis, that tumors may have other ways of doing that kind of thing that involves having extra copies of their genome. But I don't have any real evidence for that. I just say it's something, it kind of makes sense from theory. Something like that has to be going on. I'm not quite sure what it is, or if someone else in the field knows more about it than that I don't know about. Let's see, from Charles Rumberger, do you have any thoughts on the potential role of cancer stem cells in driving this process, and any differences between how cancer stem cells interact with the cell community versus how healthy stem cells interact with healthy cell communities? And again, I think it's understood to be an important part of the process, the whole story of cancer stem cells, and that to a large degree, the evolution that's happening in tumors may be happening in populations of stem cells that are then differentiating after a lot of the action of the evolution has happened. As with many things in cancers, the more we learn, I think the more complicated the picture becomes, that cancer cells are often kind of poised in this state where they're healthy cells that have become sort of stem-like in various ways and maybe have reactivated portions of stem cell programs, or they could be stem cells that have differentiated in non-canonical ways. I'm certainly not an expert on cancer stem cells, but again, it's an important part of the picture. Someone else could probably tell you more about it than I could, but yeah, I can just say that there's definitely a lot going on there that is interesting to this process, but I couldn't tell you in detail exactly how those are different. Let's see, from Adam M, thank you. Could you speak briefly to your work on optimizing sequencing studies, which I think you touched on your future work slide? Yeah, so that's something I'm very interested in. I think it's an important direction for the field that is underappreciated right now. We certainly don't have the answers there. And as a researcher, I'm kind of trying to create prototype tools as sort of a proof of concept, with the idea that if we can show people what you can do with these kinds of tools, we can convince more people to start using them, and then things will sort of take off on their own as people use them and get more power out of them. The hope would be that then more people get interested in developing them and more people interested in applying them. But as far as what we've actually done, there are really two branches of this, and one of them is simulation. So being able to simulate studies under different conditions. We've been developing tools specifically on this context of somatic evolution, so tools for simulating a somatic evolution process, but also simulating the data you would get from different kinds of studies you could apply to that process. So what would you see if you have tumor cells evolving under certain conditions, and you do a mix of bulk sequencing and spatial sequencing and single cell sequencing and different designs of how you combine those? What would you actually see? And that's one piece of it. And then the other side is the actual optimization. So this is a difficult optimization problem. It's difficult in part because it's a very high dimensional, well, not very high dimensional, but it's multi-dimensional and involves very heterogeneous dimensions. So changing things like time points at which you do time series analysis versus depth of sequencing versus different kinds of sequencing, numbers of single cells you look at. It's hard to optimize over these kinds of heterogeneous variable spaces. And it's also hard because it's very computationally costly to evaluate each possible design. The way we're doing this right now, you basically need to simulate the data, run your analysis on it, and draw a hypothetical conclusion. That's computationally costly, which makes it a hard optimization problem. So we are, for the moment, using relatively generic global optimization methods to try to solve these. I think there's a ton of work to be done on how to do this better on both the simulations, how to speed that up and get around some of the steps, not necessarily simulate everything, but simulate implicit measures, on how to do the optimization more efficiently for this domain. And I think there's a lot in the AI methods that will play into that in terms of better predicting what's going to happen without necessarily having to simulate the whole process, as well as better choosing what are likely to be the most informative data points to evaluate in these optimizations. So we are kind of taking baby steps there, but I think there's a lot more to be done. And I think that's all the questions in chat. I don't know if there's anything else anyone has in time we have left.
Thank you. Thank you so much for answering everything. You basically gave a second talk. That was awesome. I want to piggyback on the mitochondria question. I would be really interested to see that data, because we're starting to look now at mitochondria evolution as a chassis for kind of a fake synthetic cell. And it's really funny that that data is not something people look at. It seems like that should be, I don't know, naively, that should be a bigger part of looking at the whole energy metabolism of a cell.
Yeah, and again, it may be that someone has that data, and I personally haven't looked at it, so I'm not sure what's out there. I mean, I'm sure just looking at the story of clonal evolution, it's an interesting question. It's also a hard one from the phylogenetic point of view to get a handle on that. It's possible that there's work out there. I actually wouldn't be surprised if someone was asking that question, but I haven't looked at it, so I don't know what's out there.
Yeah, when I asked my friend who does sequencing, population sequencing for other types of cells, he said the mitochondria data is just trash that they're trying to throw away because it contaminates their phylogeny. So
yeah. Years ago, when I was doing more standard population genetics and algorithms for that, we were looking mitochondrial and Y chromosome are really interesting for like human population studies because they're a cleaner signal than the autosomal genome. But yeah, for the cancer case, it becomes much more complicated.
Yeah. All right. If there are no more follow-up questions, I want to say, Anselmo has a question.
Okay, so I'll read it. Michael Levin showed evidence that cell electric repolarization can keep cancer cells under control, reverting the cancerous phenotype despite their mutations. Do you think that this is a generalizable effect? I'm not familiar with that work, so it's hard to comment specifically on it. I will say that there are lots of mechanisms at work in cancers and different ways people intervene with them. So I'm not familiar with that one. But for example, cancers can adapt to, let's say, develop better motility, and then there are ways you can influence the system to try to suppress the motility and get them back under control. I don't know if this is kind of qualitatively comparable. The tumors evolve a way to be more tumor-like, and then you can intervene and try to shut that down. But I will say, in general, when it comes to bringing these things to treatment, the evolutionary process is very good at escaping all of these kinds of targeted ways of influencing a system. So you can come up with a way of suppressing a particular mechanism a tumor develops, but most cancers are very highly hypermutable. They have a high degree of heterogeneity, and it's very likely that even when you first apply that, there's some subpopulation of cells that has a way of escaping that and will just take over the tumor. So I don't know if this particular mechanism is better at escaping that, because again, I'm not familiar with it, but that's a general property of evolution in cancers, that they're really good at escaping anything you can come up with to try to attack a particular mechanism they use.
Thank you. That was, I didn't know about that work. I need to look that up because that sounds really cool.
Mhm.
All right, last call for questions. And last call. And thank you. Thank you so much, Russell, and thanks everyone. And
Thank
have a
you.
great rest of your Monday.
Thanks. Bye.
Bye.